So I'm Jack West, and a little over a decade ago, I'd founded GRACE, Global Resource for Advancing Cancer Education, and I'm very happy that it's going strong and growing more than a decade into it, and that's because of all the people in the room and some outside of it, but by that I mean both the people who are sharing the information and interested in it, and the people who are helping create it, our faculty. And today, the meeting is going to be chaired by Dr. Housekeeping from Fox Chase and Josh Baumel from University of Pennsylvania. And so I'm going to hand it over to them, but I really just wanted to thank all of our faculty for taking the time to be here, because everyone is so committed to getting the best information out to our patients directly. There are far more people who will be benefiting from this than just those who are in the room, and that is partly because several, I think, maybe still battling the cranes and trying to get in here and parked, so they will be trickling in, but especially because we are reaching a streamed audience, and this is going to be recorded, and a huge proportion of the people who will benefit from this are not here at this moment in this room, and so it's hard to imagine the virtual audience, but it's going to benefit far more than just the people here. But let me turn it over to Dr. Borgai, and thank you guys for being here.
Good morning, and thank you, Jack, and thank you for being here. We've been looking forward to this event for quite some time. I'm glad that it's come together, and we have all of our speakers coming in. And I think we really have a really nice event for you guys today. We have many experts up and down the East Coast joining us, some local folks, and some who are coming from a distance. And as Jack said, the traffic is a little bit bewildering right now. So hopefully that will all resolve and you see more of your colleagues coming in. Let me start with a couple of housekeeping items. First of all, we have Wi-Fi. So DoubleTree Meeting is the name of the Wi-Fi that you should choose. And the password is all caps GRACE19. So GRACE19, all caps, and that's for the Wi-Fi. I don't think it's a surprise to hear that events like this are not possible unless we have sponsors and supporters. I'd like to take a moment to thank our sponsors for today's event. The sponsors include AstraZeneca, Blueprint Medicine, Novartis, Boehringer Ingelheim, Spectrum Pharmaceuticals, Genentech, and Takeda. So again, a big thank you to all of them. The day is sort of jam-packed. There are many different talks separated by topics that range from why do we do rebiopsies, what's a liquid biopsy, all the way down to the nitty-gritty of how to select a therapy and what are some of the newer targeted therapies that are coming up. And I'm sure a crowd like this, most of you are familiar with a lot of these terms, but the intention is to be as informal as possible. Josh and I agreed that if you guys have a question, you can either write it down and pass it to us, and we'll read the question for the audience. You can also just raise your hand if you have a question, and then Josh and I will repeat the question just so that we can capture it in the recording, as you heard from Dr. West. So feel free to interrupt, ask the questions that you have. That's why we're here, and that's why the event is here. Most of the talks are sort of scheduled for about 20 minutes or so, but we have sort of smaller tracks also for you throughout the day, broken down by the topic and the targets that we want to discuss, and those are in the rooms to the right of the room as you exit. There's an agenda that's on the tables back there. So for those of you who are interested in following the agenda, that would be there also. So without delaying things much more, I'd like to get Josh to come up and start talking to us about next-gen sequencing. The title is Current Status, Barriers, and Emerging Targets. Josh?
Thanks, Haas, and thank you all for coming up and coming out and braving the crane. Appreciate it. Let's see. Full agenda. Where am I? I'm somewhere.
Right there, on the right, go down,
Down.
and then to the right.
Then to the right. Josh Malmo Powerball. Oh, that's me. I know that guy. Here we go. All right, here we go. So, as Haas said, the goal is for this to be informal. If there are questions that arise, you can call out, you can raise your hand, you can throw a paper airplane at me, but your aim has to be very good, because if it goes over there, I can't answer the question. So I'm going to talk about next generation sequencing and really try to give a 10,000-foot view of where we're going to be, the topics we're going to be discussing today. We're going to be able to, I'll give a little sort of brief blurb on some of the topics that you'll get longer talks on later in the day. So lung cancer has really undergone a major evolution in the way we think about it and the way that we treat it. It used to be this major monolithic entity. Patients would be diagnosed with it, and then unfortunately the outcomes were poor, universally poor. The first advance came based upon histology. So pathologists looking under the microscope at cells and saying, okay, these cells are small, these cells not so much, and that's how we get non-small cell and small cell. But breaking it down further, these cells look different. These are adenocarcinoma, these are squamous cell, and so we would give them targeted treatments as a function of that. But still, it was rather blunt in terms of our approach. And here's where we are now. And this is just looking at adenocarcinoma, okay? And what this represents in this pie chart here is we have each type of lung cancer is a piece of that pie chart. And what it means is that if you take a look here in the green, you see EGFR. That's the most common targetable alteration seen in non-small cell lung cancer. But if you see these ever-shrinking pieces of the pie, what it means is that if a patient goes in to see an oncologist in the community with a new diagnosis of lung cancer, there is a very real chance that that patient will be seeing an oncologist who has never seen that type of cancer before. And that is why organizations like GRACE are so important, because it's important that somebody is aware of what is out there. And as oncologists treating everything, it can be really overwhelming to be aware of all the aspects. But when it's your cancer, it's something that you can really focus on, and there are opportunities through GRACE to learn about this. The other aspect, which I'd call your attention to, is on your right over there, the blue unknown. Does that mean that there are no other targets that we can use drugs for? No. It just means we haven't figured it out yet. And so we have a lot more work to do to make sure that treatments that we give for patients with lung cancer are as personalized as possible. So this is, when I was a kid, what I thought mutations represented. These are the X-Men, and I thought that this was what a mutant was. I thought that if someone had a mutation, they would be able to shoot beams out of their eyes. There would be lots of cool special abilities that came with it. I went to medical school, found out this is not true. I don't want to bust anyone's bubble about this, but so.
All the way to medical school?
Yeah, it took a while. Anyway, I'm a bit slow. So anyway, so there are words that we use in medicine, though, and as doctors, we use big words. We really like big words. And I think that it's really important to know what they mean. So the first one, whenever I tell a patient that they have a mutation in their cancer, the first thought that I usually get from them is, what about my children? What about the other members of my family? So most mutations that are seen in lung cancer are what we call somatic, meaning they occur in the tumor. They are not what we call germline, meaning they can be passed on to other family members. And generally speaking, they are completely homogeneous in that tumor. So if an EGFR mutation is present in that tumor, every single cell of that patient's tumor has that EGFR mutation, and cells in their body that don't have a tumor, a normal healthy liver cell, for instance, would not have that mutation. most of the alterations we see are either called a mutation or a translocation. And the other word I've heard used when we're talking about translocations is fusion. And I actually find that talking about it from this perspective can be helpful in terms of pictures. So if you see over here, these are 2 chromosomes, okay? Chromosomes are what we keep our genes on. And a mutation means that if you look over here, this blue one, it was blue, blue, and now all of a sudden green. And the body sees that and says, okay, that's not normal. And a change can happen in the protein that that gene codes for as a result of that change. And the other type of mutation that can happen is a deletion. Now, I know we use a lot of medical words, but deletion, most people understand, right? A piece of it's missing. So you see how this red one over here was the same size. Now a piece of it's missing. So if you imagine that there was a gene that made cells grow, and there's a part of that that says, hey, stop it. If that part gets deleted, the cell will continue to grow, and that's what cancer is. The other thing which we talk about are translocations. That's where the chromosomes break, and part of them get stuck on different parts of the chromosomes. And so this is what we can see over here. Here we can see that part of the blue one ended up on the red one, part of the red one ended up on the blue one. And this can make different proteins behave differently. Each gene has an on and an off switch, essentially. And so you could imagine that if there was a gene, for instance, that made your fingernails grow, you'd be okay with that being on all the time, right? It's okay, you can clip your fingernails, it's not a big deal. But if that same on switch was attached to making cells grow, that would be highly problematic. So this is sort of the normal conditions of what we would call, the on-off switch, right? We have a light bulb, right? We have an on switch, there's a promoter, it turns on, light goes on, you turn it off, light goes off. So an oncogene, something like EGFR, epidermal growth factor receptor, is a gene that when activated makes cells continue to grow and grow and grow. And what you see here is that you turn on the off switch, it's not turning off. And yet for things like EGFR and BRAF, we have a targeted therapy that we can administer that essentially will block that function of that protein, and that's like unplugging the lamp. Right? So that we have a backdoor that we can prevent that from causing a problem. Tumor suppressor genes, things like P53 and RB, are somewhat more problematic. So when what those are, they're off switches. So P53 turns off lots of genes, and what we can see here is that, it doesn't matter if the light is plugged in or unplugged, it's going to stay on. And the other problem is that this affects multiple pathways. It's not just unique to one spot. So when a tumor suppressor gene is altered, it's more difficult to target it. And that's why even though P53 is the most commonly mutated gene in human cancer, we don't have targeted therapies that lead to improved outcomes when we target it. So why is targeted therapy important? I don't need to tell this room that, but I think it's important to see a picture. Right? So this is from the original paper that evaluated crizotinib for ALK translocated non-small cell lung cancer. And what we see over here, this is a patient had metastatic disease. You can see this is the patient's left lung. You can see this is the disease in the lung over here. And then at the first scan after treatment, you can see that all of that had melted away. And that's what we tend to see with these targeted therapies. When a patient has the target, and you give them a pill that targets it, we see rapid and durable responses. So how do we test for this? Historically, we used to use things called PCR, polymerase chain reaction, IHC, immunohistochemistry, or FISH, fluorescent in situ hybridization. All of these are still used because they're fast, but the problem with them is that they only look at one gene at a time. And when we consider the fact that we have multiple genes we're interested in, every time we do a test like this, we use a little bit of tumor and a little bit of time. And our biopsies are small, and patients with lung cancer don't have a lot of time to be waiting for these things. And so that becomes problematic because the way we used to do it, we say, okay, do they have BRAF? No. Okay, do they have EGFR? No. Okay, do they have ALK? No. And each time we do that, it takes time. Okay? So this is how we're doing it now. It's called next generation sequencing. It's a little bit of an unoriginal name, because what are we going to do when we have our next sequencing? What are we going to call that? But for now, we call it next generation sequencing. And so the issue is not so much anymore tissue and time, because we only need a little bit of tissue, and in parallel, we're able to sequence hundreds, sometimes thousands of genes at the same time to get information. The problem becomes the bioinformatics. When you have a panel that spits out information on 1,000 genes, and you only have targets for 5 of them, what are you going to do with the information? How are you going to affect clinical decision making when you see these other changes? When I first started doing this testing, my first patient I sent it on had an androgen receptor mutation. That's the male hormone. Androgen receptor mutation in their lung cancer. I remember looking at this. I have no idea what that means. And the patient asked me to say, well, what should we do with this? And the truth is, at that point, you shouldn't do anything. But it's important to find these targets that actually do have very effective treatments. It gets more complicated, unfortunately. Most next-generation sequencing panels that we use utilize DNA. So to go back to biology class, in the cell, all the genes are encoded in DNA, which is double-stranded, which you can see here. In order to be made into a protein, first it is turned from DNA into RNA. RNA is single-stranded. And then, once you have the RNA, first it's pre-RNA, then you cut out parts of it that aren't actually coding for anything. These are called introns. Now, why am I going through all of this? I'm going through all of this because as a result of this process of cutting out the introns, traditional DNA-based NGS sequencing is not very good at detecting translocations or fusions, because these introns can be really, really big. And you could imagine that if you were walking along a path just following breadcrumbs, you'd say, okay, this looks fine, this looks fine, this looks fine, this looks fine, and you could end up on a totally wrong path because you're just focused on the one gene in front of you. And when big changes happen in the introns, which are areas we're not really focused on, it can be difficult to notice that. So RNA-based sequencing, which it's not technically next-generation sequencing, it's a multiplex PCR, but you'll forgive the simplification, is much more useful for detection of translocations. But it's not done as frequently. I have had many patients who have testing, their tumor tested, and their oncologist will say, oh, the tumor was tested, and they come to see me, and I find, actually, this was not tested for translocations. The DNA-based NGS assay you're using cannot detect an ALK translocation. In the last year, I've had 3 patients who I found ALK on who were told they had no targets. And it's very important to look at what testing was done so that you can give the right treatment. So who should be tested? Anyone with metastatic adenocarcinoma of the lung should be tested. And even though these alterations are more common in never or light smokers, we have definitely seen these changes in patients with a tobacco history. We should not be judging patients based upon their tobacco exposure. In addition to that, if someone has squamous cancer, but they've never touched a cigarette, they are, a very young person, I don't trust that diagnosis. And I will still do a sequencing panel on that person. I'm sure every oncologist in the room has seen patients with squamous cancers and an EGFR mutation, or squamous cancer and an ALK translocation. And if you want to think about why, when we do the biopsies, I know they are difficult and painful and traumatic for patients, but if you think about the size of a tumor, and then what they're actually putting in is a tiny needle. So you have a sphere, you put a tiny needle into it, what you get is what's in that spot. And if that spot looks a little squamous-y, you can be called squamous. But it's important to think about the whole clinical picture. At this time, NGS testing is FDA approved one time for any patient with cancer. That decision's a little bit problematic, but we're not going to talk about that right now. the end result is that there is no reason why a patient with adenocarcinoma of the lung should not have rather comprehensive testing. There are currently 5 genes with FDA approved therapies for patients with such alterations: EGFR, ALK, ROS1, BRAF, and NTRK. And really, it's important to ask your oncologist, say, find out what genes were tested and how were they tested. You can ask, was NGS performed? Does the assay that was used detect translocations? And these are the questions that you can ask. They should not get offended by these questions. These are appropriate questions that a patient should be asking so that you can get the appropriate treatment. And new targets are coming. So I showed the ones that have FDA approval. This is RET translocation data with 2 emerging drugs. So this is a waterfall plot. For those who don't spend all of their lives reading oncology journals like I do, each of the patients who are on this trial is represented by one of the columns in this plot. And so what you can see is that if the column goes down, it means the cancer got smaller. And if the column goes up, it means the cancer got bigger. If the column passes this little dotted line, that means that we call that a partial response because it's over a 30% reduction. What you can see is that when patients with RET translocations were given either of these drugs, which still have what we call license plate names with a bunch of letters and numbers, they don't have names yet, really good things happen. And so it's important to understand that there are targets that are not yet FDA approved for which we have trials, for which we have really exciting new outcomes. And if you look at the toxicity, which is indicated over here, these are treatment-related adverse events, you see that these drugs are really well tolerated. So RET translocation is one that I tell a patient, if someone called me from, I don't know, I'm not going to pick on any particular state, but a state where they are not near an academic medical center and they have a RET translocation, that is a patient I tell to get on a plane, okay? Because there are very exciting trials that we can get for that. And so knowing what you can treat with is really important. So the key points to remember, though, is that at this point, unfortunately, most patients do not have a target, and yet knowing the target for those patients who do makes a really big difference. It's a very big difference taking a pill once a day as opposed to coming in for infusional chemotherapy. Those patients who do have a target should receive the targeted therapy. The response rate is better, quality of life is better. If we need to start treatment before next generation sequencing comes back, because sometimes it could take about 2 weeks, if a treatment's working and they're tolerating it okay, we keep going. But it's very important to get this information as soon as we can. Resistance can be heterogeneous, and this is why repeat testing is important. So looking at mechanisms of resistance is very important. So I believe that Haas is going to talk about repeat biopsies and why those are relevant for patients with lung cancer receiving targeted therapies. Getting that repeat biopsy can be very difficult. So one of the emerging topics that Dr. Agarwal is going to discuss is about liquid biopsies, how we can incorporate that, so drawing a blood sample to get some of the same information. This is just looking at what a cell-free DNA or liquid biopsy looks like in a graphic format, but Dr. Agarwal will talk to us more about that. And the other thing which we're going to talk about later is that you're not alone. So there are multiple advocacy groups and patient support groups for various of these targets that exist. And I think this is important because if you're told that you have a lung cancer that is present in only 1% of all cancers, it can feel really isolating. But in the modern era, you do not need to be alone in this process. So I'll thank you very much, and I'll take any questions.
Well, we will have time at the end for a panel, but just your impression of one of the issues, and you just touched on a bit of that what we have now is just a fraction of what is coming. And
yeah.
This has been a moving target for years and will continue to be. But how much of a problem do you see for the ambiguity of what is actionable and should change treatment, particularly when we get reports back that list a lot of mutations that may or may not have results that should change our practice?
I think that those
I'm sorry,
reports.
Can you summarize that question?
Oh, absolutely. I'm sorry. I promised I would do it.
Yeah.
So Jack's very good question is, if I may summarize it, is that when we send next generation sequencing assays, the reports that come back will have huge numbers of mutations on it. And only the minority of them are actually clinically actionable. And how much of a problem could it be for patients or oncologists to treat based on those less than slam dunk targets? And what should we do about that? How much of a problem? Did I capture that?
Particularly the gulf between you at an academic center and folks who are way out there who don't have access to that.
Right. And so for me as an academic oncologist, all I do all day, every day is lung and head and neck cancer. So I can think about this stuff and I can say, oh no, that target didn't do well in this small study we saw at a poster in some city where they weren't there, right? So like that is a benefit that I have as an academic. But I think that the other key problem, if you guys have seen these reports, is they will say mutation, and then they will say drugs that might work in this situation. And that column drives me insane, okay? Because it will say things like, P53 mutation might respond to these drugs, and I'll read it and say, no, it won't. That is based on nothing, but that's cool. And they'll say things like KRAS mutation, for which we, unfortunately, this time have no targets. And they'll say, oh, you might want to give an mTOR inhibitor. And an mTOR inhibitor is, I guess, biologically, hypothetically might help, but is really toxic and has not been shown to be effective multiple times. So I tend to ignore that section. And I think that it is a problem. But I think that the benefit of next generation sequencing outweighs that. And the other factor which I will say to these tests' credit is if you've ever seen the raw data that they use to make this report, it will give you nightmares, okay? It's a bunch of letters and numbers in a row. It really looks like nothing. And that they come out with anything that's clinically meaningful is very useful. So I think that the key thing is to make sure that we approach this with caution. And to a more real world situation, if a patient has resectable lung cancer and we find they have an EGFR mutation, that's something where we have a target, right? I know I have a target, but it's not FDA approved to use a targeted therapy in that setting. And I think that we need to be diligent. Say, we don't have data to say it helps here. I will have this in my back pocket as another option, but still I'm exposing someone to a treatment that has potential side effects, and I should not do that unless I have data to indicate that I'm going to improve patients' outcomes. And so that's really the way I view it.
So at academic institutions, you do next generation sequencing. What about other places? What type of sequencing do they do, and should patients be asking, and what should they be asking for?
So it's highly heterogeneous. So the question was, at academic centers we're doing next generation sequencing, what are they doing in communities? What should they be asking for? It's a fantastic question because most oncologists, when I travel around and talk to people about this, and I'll say, well, what testing are you doing? They say, oh, we do testing. And the reason for that is that when doctors go through training, we get specialized, focused training. We learn about I learned about internal medicine, then I learned about oncology. I didn't learn about how to run a lab and how to run a PCR test. And so we rely on our colleagues to do that. And so we say, well, I sent off the test to the pathologist and they did the testing. Most next generation sequencing that is done outside of an academic medical center uses a commercial assay. Foundation Medicine, Blueprint Medicine, Caris, any of these assays can be used in that setting. But the key question which you should ask is that some centers are not doing any next generation sequencing, or they're worse, not doing any testing at all. And so the patient should say, what testing was done? Can I see the report so we can figure out what type of test was done? Because even though it's intimidating, at the bottom of the reports, it will say, we did this test. This is what this test can find. And I think that's useful. And patients can be intimidated to ask their doctors something like that, but it's important.
Guys, why don't we do one more question, because then we're going to be.
Yeah, I don't want to push on everyone else's
We have
time.
panels,
We have a
discussion also.
great time for question and answer. One more question?
Okay.
Yes.
This is a quick one. So the commercial assays like Foundation One, Guidant, are those RNA or DNA sequencing?
So the question is about commercial assays, whether they're DNA or RNA. The only assay that is commercially available that I'm aware of that includes both DNA and RNA is MSK Impact. Their foundation at this time is DNA based, and I believe the others are RNA. Is there another one that's RNA based that's commercial? I don't think so. But like at Penn, we have our panel. We have a RNA based Archer assay, which we send every patient with adenocarcinoma is sent off for our DNA panel and our RNA based fusion panel. All right.
Thank
Thanks.
you.
Thanks, Austin.